Hello everyone, this is Seher from EasyPeasy, and the topic that we are going to discuss today is called as the cell cycle or cell division and its regulation. Cell division usually occur for certain events, just like reproduction is one of the events when cell divides rapidly to make a new being, or growth of an individual from a baby to an adult, or the regeneration process. Just like here, the starfish is going to cut itself into two pieces and then each piece is going to regenerate itself. Or our hair cells or skin cells and nail cells are constantly regenerating. All these events involve the process of cell division. And cell division usually occurs in all types of cells, whether it's a prokaryotic cell division or a eukaryotic cell division. If it's a prokaryotic cell division, then they are usually called as binary fission. We will make a separate video for a binary fission because if we include binary fission in this video, then this video will get too long. And in eukaryotic cell division, we have both animal and plant cell in which the cell divides. Now, these cell division usually take place in a series of stages, and these series of stages basically called as cell cycle. Now, the cell cycle have two main categories. One is the resting phase that is called as interphase, and one is the dividing phase and that is called as M phase. In M phase, we have mitosis and meiosis depending on the type of cell that we are dealing with. If it's a normal cell, then the process will be called as mitosis, and if it's a germ cell, then the process will be called as meiosis, followed by a little part in which the cytoplasm of the cell divides, and this part is called as cytokinesis. Okay, now let's start with the interphase. Within interphase, we have three subcategories. The first category is called as G1 phase. G1 phase is basically a phase in which the cell contents, excluding chromosomes, are duplicated. So all the things that are needed for a cell to divide, for example, all the ribosomes, mitochondria, Golgi complex, etc., will get duplicated in this phase, followed by the next phase that is called as S phase. S phase is basically a phase in which chromosomes get duplicated. So in humans, we have 46 number of chromosomes, so they will get duplicated in this phase. After S phase, the last phase of interphase is called as G2 phase. G2 phase is basically a type of checkpoint for us in which the cell double checks that all the chromosomes are without errors. And if there is any repair needed, then the repair will be done within this phase, and then the cell will get ready for nuclear division. Nucleus divide in the M phase, followed by the division of cytoplasm in the cytokinesis, and in the end, we will have two daughter cells. Sometimes, due to some reason, maybe the cell is too old or have some kind of damage and the cell is not able to divide further, then they will go into G0 phase. In G0 phase, the cell cycle arrests. Okay, now let's start with the M phase, and we will specifically talk about mitosis in this video. For meiosis, we will make a separate video for that. So in mitosis, just like interphase, we have four subdivisions. That includes prophase, metaphase, anaphase, and telophase. Now let's talk about each phase in detail. So we will start with interphase, specifically the G2 phase, in which all the things are duplicated. So you can see there are two centrioles here, and we have a nuclear membrane. Nucleolus is here, and the chromatin material is spread inside the cell, and there is no visible chromosome. Once the cell enters the M phase, it will enter the prophase. In prophase, the centrioles are going to get apart like this. Nuclear membrane disappears, nucleolus disappears, and chromatin material condenses itself into visible chromosomes. If you look at this chromosome closely, you can see that the chromatids are joined tightly with each other with the ring type of structures here. These ring type of structures are called as SGO1 bound cohesion. And near the centromere, where we have the inner kinetochore and outer kinetochore, we have these red rings that is called as CENPU bound cohesion proteins that are going to bound these two chromatids tightly with each other. Once this prophase enters into the metaphase, so we will have a middle phase called as prometaphase. In prometaphase, all these SGO1 bound cohesion proteins that are these blue ring type of structures will go away from these chromatids, and the chromatids will look like this. So as you can see over here, all the blue rings are going away and the little is left in the middle. We can also see the green microtubules are getting out of the outer kinetochore tubule of these chromosomes. Once this prometaphase enters completely in the metaphase, we can see that the nuclear membrane completely disappears at that point, and the centrioles will make spindle fiber. And all these chromosomes that were scattered inside the cell will going to arrange themselves in the middle of the cell. At this point, all the SGO1 proteins will remove from the chromosome, leaving the both chromatids far apart. Now, this spindle fiber that is originating from centrioles are not that simple. We have different types of microtubules originating from the centrioles, making it a spindle fiber. So the first microtubule that we are going to talk about is the kinetochore microtubules, that is represented with the solid lines. These solid lines are going to attach themselves with the kinetochore tubules that are coming from the chromosomes. Then we can see the dotted structure microtubules, and they also have charge on it, and these microtubules will be called as polar microtubules. They're not attached with the chromosome, but they do have a charge on them. Then we have a third type of microtubules that are overlapping with each other like this, that is represented in red color, and they will be called as overlap microtubules. And last but not the least, there are some microtubules, as you can see on the side of the centrioles in green color. They will be called as astral microtubules. And as we discussed before, that the green microtubules are also originating from chromosomes, so they are overlapping like this. Over here in this picture, this green microtubule is coming from the chromosomes, and this brown microtubule is coming from the centriole. And they are overlapping just like in the movie of Avatar, where people were making bonds with their horses and dragons with their hair. Now let's discuss the functions of all these microtubules. Astral microtubules, as these are the tubules that appears initially, work as an anchor for the cell. When the spindle formation occurs inside the cell, it makes sure that all the chromosomes will align themselves in the center of the cell in metaphase. Then we will talk about the overlap microtubules and polar microtubules. Overlap microtubules are also called as interpolar microtubules. And during the phase of anaphase, these microtubules will elongate, providing the capacity for a cell so that the cell can divide easily by this process. On the other hand, the kinetochore microtubules that are attached to the centromere of each chromatid are going to shrink. So they will shrink and take that sister chromatid that is attached to each microtubule towards their pole. Okay, so these are the functions of microtubule. Let's discuss how they will apply their functions and get inside the anaphase. So in anaphase, firstly an enzyme will appear that is called as separase. This separase is going to cut the red ring type of structure that we discussed before, and these structures were CENPO bound cohesion proteins. They will degrade that cohesion proteins, and now the chromatids can go towards their pole easily. Next, as we discussed before, that polar microtubules are going to elongate and kinetochore microtubules are going to shrink. As a result, the anaphase will look like this. Now here, as you can see, all these sister chromatids are towards their pole. You can see that the polar microtubules are elongating and the kinetochore microtubules are shrinking. Now, the next phase is the last phase of mitosis, and that is called as telophase. In the telophase, the first thing that's going to happen is the reappearance of nuclear membrane. And the chromatids are going to disperse themselves again in the form of a thread like this. And nucleolus will also going to reappear. So now the cell division is almost done, and now it's going to enter the cytokinesis phase. In the cytokinesis, as we discussed before, that the cytoplasm of these two cells are going to divide. Now, the division of the cytoplasm depends on the type of cell it is. So if it is an animal cell, then there will be a ring type of structure appears that will be called as contractile ring. This ring will initiate the division of cytoplasm of the two cells. And if this cell is a plant cell, then there will be visible plates that will appear in the middle of these two cells, and they will be called as cell plate. These plates will be resulting in the division of cytoplasm into two cells. And then in the end, we have two daughter cells after mitosis. Okay, the next part of the video is the regulation of the cell cycle. On this cell cycle, we have certain different points where the regulation will be done before entering into the next subcategory. These points are called as restriction points. They can also be called as checkpoints. So the checkpoints that are usually present on the cell cycle are M1/G1 checkpoint, G1 checkpoint, G1/S checkpoint, intra-S checkpoint, and G2/M checkpoint. There might be more checkpoints on this cell cycle, but these are the most common or most prominent checkpoints that are discovered by the scientists. Okay, now this regulation will be done by an enzyme, and that is the cyclin-dependent kinases or CDK. When you hear the word kinases, it means this is an enzyme that is going to attach a phosphate group on its substrate. And it is cyclin dependent. It means that there will be a protein named as cyclin that is going to attach itself with this enzyme in order to activate this enzyme. Okay, now let's discuss this cyclin dependent kinases in detail. So this is our enzyme, that is the cyclin dependent kinase. You can see the structure of this kinase like this. So there is a dip over here, okay? Now this is cyclin dependent, so a cyclin protein will appear that is going to attach itself with it like this. And now, as you can see, that the structure of the CDK is changed, making a depression here, and this depression is basically the active side of this enzyme. So now this enzyme is activated and it is ready to attach itself with its substrate. So when a substrate is going to attach itself with this enzyme, it needs the energy in the form of ATP. So they are going to attach themselves with the enzyme like this. And as we discussed before, that it is a kinase, so it is going to attach a phosphate group with it. And where this phosphate group will come, this phosphate group will come from this ADP molecule. So ATP converts itself into ADP, attaching a phosphate group to this substrate, and then they can go away from this enzyme like this. Okay, so this is the general idea how the regulation of each checkpoint is gonna be done. Now let's discuss specifically with one restriction point and see how they are going to regulate that checkpoint. So the checkpoint that we are going to discuss right now is the G1/S checkpoint. Now, for example, the cell is sick and it cannot go into the next subcategory of the cell. So at this point, there will be a protein named as retinoblastoma protein that will get activated. Now this retinoblastoma protein is going to halt the process at this step and will not let it go to the next subcategory. How it's going to do it? Because it's going to attach itself with E2F protein that is attached with the DNA. For example, the cell repaired itself and now the cell is ready to go in the next step. So at this point, what happened is that the cyclin protein will get generated, that is the G1S cyclin protein. And this protein will attach itself with the CDK enzyme again like this. And this retinoblastoma with ATP is going to attach itself with CDK enzyme. And now it is going to attach a phosphate group to this retinoblastoma. And this retinoblastoma that is phosphorylated is not able to attach itself back to E2F protein. So that's how the process of cell cycle will go from G1/S phase back to S phase. And once it passes through this stage, the G1S cyclin protein is going to get degraded itself like this. So for MG1 checkpoint, we have cyclin D protein attached with CDK4 or CDK6. G1S checkpoint will get regulated by cyclin E protein with CDK2. Intra-S checkpoint is going to get regulated by cyclin A protein with CDK2. And last but not the least, the G2M checkpoint is going to get regulated by cyclin B protein with CDK1. So that's how the cell is going to get regulated on each and every step. Even at the time of metaphase, when this CE and PU protein is going to get degraded, the cyclin-dependent kinase is going to work itself over there too. So as we discussed before, that in anaphase, we have an enzyme called as separase that is going to degrade the CE and PU bound cohesion protein. This enzyme will get activated when we have the M-cyclin protein attaches with CDK and this CDK attaches with APC. APC is basically anaphase promoting complex. And as a result, this separase is going to degrade the CENPU protein like this. And now the chromatids are free to go towards their pole accordingly. That's it for now. The notes and little quizzes for this video will be available for EasyPeasy expert members. Thank you very much for watching this video. If you like it, please subscribe this channel. Thank you. Bye for now.